Dr. Anoosha grew up in India, did her bachelor's and master's in bioinformatics degree of technology in Chennai, India. She received her PhD in protein bioinformatics with special focus on cancer under Professor Michael Gumia at Indian Institute of Technology, Madras. In 2018, she joined the Roy Choudhury team at Ohio State University as a postdoctoral researcher. Her research interests are to study human heterogeneity and evolution in different cancers and develop computational methods to assess heterogeneity and find distinct cancer cell populations in a tumor that try resistant to drugs. We welcome you, Dr. Anusha. Thank you for joining us. You may share yours.
Thank you, Sunita, for a nice intro. And also, I would like to thank Medha and the team for inviting me for this talk. And also, I would like to thank Dr. Elson for a wonderful talk about machine learning algorithms. Actually, that made my job easy now. So I don't have to go through the basics of machine learning as I walk you through the slides. I will sharing my screen. 1 second, please. So I am Anusha. So my research domain is specifically focused on cancer. So I'm trying to answer some of the questions that are to be solved, and like some of the unsolved puzzles in cancer research that ultimately lead to patient care. So I would in this particular talk, I would like to focus more on applications or like roles of AI in cancer research, since my area is like cancer. So please don't hesitate to stop me anywhere in between if you are not familiar with any of the terminologies which I use that are related to biology or cancer, whatever. So I prefer stopping me in between the presentations rather than asking some basic questions at the end without following the presentation. So please feel free to ask questions. Starting with some of the basic questions, like what is AI and like what are the technologies within AI. So Dr. Elson wonderfully explained all of those. So I'm not going into that question like what is AI and all. So now by now like you all are familiar with that. So I think like the main focus of this talk, I would like to talk more about the scope of computational biology, especially AI in cancer research. So to begin with, so before I jump into the topic, I would like to give a brief info of like what is cancer. I'm not sure like how many of you are like very familiar with the processes happening in cancer, but just a brief intro, like what is exactly cancer and what we call a disease as cancer. So usually in a normal human being, what happens? The normal cell division happens. The cells keep multiplying. So the picture here, the picture above the line here represents the normal cell division in a human being. So this normal cell keeps dividing by many like process called mitosis. So what happens when a cell damage happens in between the natural process? So some like most of the times, like what happens is there is a wonderful repair system in our human body that will repair the damage that happened to the cell, and then the cell continues as the other normal cells. But in some cases, what happens unfortunately is the cell damage isn't repaired, and the damaged cell keep dividing, and then that ultimately leads to abnormal growth of cells, so that we call as cancer. So now like I'll be discussing about like AI, particularly to this cancer. Where can we apply this AI technology for the patient care who are suffering with cancer? So when I was looking into the literature about AI for healthcare, specifically for cancer, so I found this like image, which is like very how to say, it's like a brief summary of different levels of patient care from the start of diagnosis of the disease until the end of the treatment or whatever applies, end of the life care. So here, so in cancer perspective, so I think like early diagnosis is the key, right? Like early the diagnosis, the better the treatment. So that the clinicians can suggest a better treatment if we detect the cancer like very early in its stage. Like how AI comes in here. So I think like there are AI methods here at each and every step given in this figure. So at the stage of diagnosis and at the stage of decision making, like what kind of treatment we given to the patient and improving the treatment patient as the treatment goes on, and the other aspects of patient care in the clinical setup. So so I think like I'll be focusing more on these topics here I listed on this slide. So how can AI help see cancer in new and better ways? So I think like, there are like multiple stages of patient's treatment, and like at all of these stages, like we can apply AI to improve the patient care. So first thing, like I said, like diagnosis, which is the key for any disease, like even in cancer, it's like the key. Mm. So coming to AI in cancer diagnosis. So how does the cancer is diagnosed for a given patient? So patient approaches the doctor. So with multiple complaints, right, like different symptoms. So based on the symptoms, doctor suggests the scan. I think the scan is the first step in the patient care for the diagnosis. So the doctor suggests scans to the patient, and they undergo the scans based on the symptoms. So the scan could be any organ, like again, based on the symptoms. So here, this slide gives an example of a brain tumor patient, glioblastoma. So the figures here represent the scans of the brain, MRI scan of the brain. So here, let's see how AI is helping to improve the diagnosis of brain cancer. So the figure in the left, so both of these figures are the brain cancer images. So the figures in the left are a different type of brain cancer, and the figure in the right is again a different type of brain cancer. So when you look at the left side of each of these figures, so the left side of the scans are the scans, are the MRI scans of the brain, and the red ones which are highlighted here are the tumors. So it's like very hard for anyone, any human eye to distinguish the tumor tissue from the scan unless and until the physician is like very experienced looking at the scans of this cancer type. So whereas we feed in this information, the human knowledge into the computers, so the right side of the figure represents the output of an AI algorithm, which is a convolutional neural network, which is an unsupervised deep learning method. Like I'm not going to into details of these methods here again. So what does the AI did here? So it almost like identify the tumor, which is here highlighted in red and yellow in both of these images. I hope like you can see my mouse moving around these figures. Sorry, like
Yes.
not okay.
Okay.
Like AI like almost identified the tumor tissues here from these images, almost close to what the clinician has identified from the scans. So in this way, I think like AI is helping to make the process faster, and also like it is also helping in a setup where there are like very less experienced radiologists or clinicians with these images, right? Like this is definitely helping to fasten the detection diagnosis part of the patient and improving the patient care at this step at this step particularly. So not only like for diagnosis, not only cancer imaging, like not only the CT scans, there are like multiple methods for how to say for diagnosis of cancer. So I think like so the first step is CT scans, and then what happens is if they find any suspicious tissue, abnormal tissue growing in the scan, So the doctor suggests them to undergo the biopsy. So biopsy is something like very painful, invasive method to collect the tissue from the samples. And I think like it's really a painful method to perform on already a suffering patient, right? So I think like AI here, if it is like much more reliable, I think like we can, like I'm not sure like if it is right now going on in the clinic. I'm not like, I don't think it's going on, but we can avoid the biopsy step, like we can avoid the patient from suffering collecting the biopsy samples. So in case if we collect the biopsy samples from patient, so the samples are being like will be processed by the experimental biologist in the lab, and then the samples go to the pathologist. So the very well experienced pathologist, he will look the slides, the tumor slides, with the cells laid on through the microscope and see the histology of cells, and again try to estimate the cancer volume or the whatever, like different parameters that are defining cancer. So that is the second step. So even those slides are very difficult to look at through the microscope and estimate all these parameters with human eye, right? And it is also like very tedious job for any pathologist. So again, here comes the application of AI. So again, AI can help human brains to quickly identify the abnormal cells and like what type of cells in those cancer patients and all those parameters. So that is one step where AI plays a major role. And another thing is like from these images, like from the scans, we can also predict the different stages of cancer. So when the patient comes to the doctor, so unfortunately, it is always like mostly at the later stages of cancer. Unless it is in the later stages of cancer, the patient doesn't know he is having it. So he or she is having it. So I think that is like very unfortunate thing. But like when the patient arrives to the clinic with the scans, so AI again can help here to predict the stage of the tumor, the stage of the cancer, which again makes it easy for the doctors to to decide the treatment to be given to the patient, and we can in that way we can fasten the patient care. And one more thing is like, so I'll be talking about the associating these images with the genomic data and monitoring disease at different timelines of treatment in my coming slides. Sorry, like you can stop me any time. Like any questions so far?
There is one question in the chat box if you want to address it now. So someone asked like, can someone use AI in brain modulation and neurology diagnosis?
Uh-huh. So to answer that question, like I don't have any experience on analyzing these scans, but definitely I think the answer, like simplest answer to that is yes, because like there might be like different kinds of patterns in the images uh, to understand, right? Did you get me? So
I think okay.
there is always like a image from the normal brain, right? Like we can always use that as a reference and compare the one which is abnormal to the reference one. So that is how AI works, right? Like if in case in supervised learning, so we feed in the known data to the method and it recognizes uh, which one is odd from the normal one, right? So I think the simplest answer is yes for that.
Yeah, please go ahead.
So I think like I discussed about the diagnosis part, which is the first step in the patient care. So in the later stages, I'll be jumping into the gene level of this cancer. So this is my expertise here. So I work with lots of next generation sequencing data. So to make it easy for everyone here to understand, I'll just simply like in simple terms. So what we do with the tumor samples. So on a daily basis, we collect the samples from tumor patients. So sample is nothing like in a layman terminology, I can say a small amount of liquid in a tube. So we get it. So that is a tumor sample, or it can be a tissue, a chunk of tissue from the patient. So it can be either ways. So we get the sample to the lab, and the experimental biologists in our team, they process it in such a way that we can sequence it. So when I say sequencing, so what does that mean? So we send the sample, like sequencing is nothing but we are trying to understand the genetics behind the tumor sample, like what's happening exactly. So what does NGS do? So at each of these steps here, AI plays an important role. So, and again, to remind all of you, like AI in all of these steps are still in research, and I don't think any of them are any of them have crossed the research lab and entered the clinical setup. So all of this is like undergoing like in process of research. So NGS technology. So we send the samples for sequencing. So all those machines out there in the market which do the sequencing are like some of them are again AI trained machines. So they read the sequence. So our how gene sequence will be there in the sample, right? So each of these human cells have a string of letters. That is what we call genes. So what we get out of those machines is nothing but a string of 4 letters. So all our genes are coded by like just 4 letters. We call them bases. So it's they all are set up in a meaningful way that they code to genes. Uh, so we get these sequences. For example, here what I represented are like CATG. I think like before we have 4 CATG letters in the gene. So 3 of these bases together form a codon. Each of this codon again represents a single amino acid in the protein. So I think the final functional unit of the cell is protein. So our cells function based on the functionality of those proteins in the cells. So what happens when we send the sample? So we get the strings of letters. So it's not as easy as I show it here, but like we get terabytes of data out of the machine for one sample. So I think the role of computational biology in research setup comes here. Like how are we going to analyze this data here? So we get these strings of letters, we then process this. So we get these strings of letters, we compare it with the reference strings of letters you call genome. So the entire set of strings of letters, we call it a genome in a human body. So we compare these abnormal letters with the reference genome and see what happens. So I like when I do this alignment, so this is the top one is the reference one and the below one is the abnormal sample, the tumor sample. So I see a particular change here. For example, the letter A has been changed to letter C. So that ultimately codes to a different amino acid. So before these 3 letters together was coding to another amino acid called histidine. And then because of this alteration here from A to C, the codon is now coding to a different amino acid, a proline. So in that case, what happens, this single amino acid change might produce a non-functional protein, or it might not have any effect on the protein. We doesn't know. So we just see the alterations here in this step. So we call that variant calling. So again, in most of the cancer types, I think the prominent alteration which we see is the single nucleotide change, the single base change. And there are like many other types of alterations seen in this genome, like in the tumor samples, but the most prominent ones are the single base alterations. So we call this step as variant calling. So there are many algorithms out there in the literature to do this, to solve this problem here, variant calling. So there are also again many AI methods, machine learning algorithms to call variants from the given tumor sample. And also there are like many machine learning algorithms to align the tumor genome to the reference genome. So it's like, I just wanted to highlight like different steps, and each of these steps have a place for AI. So each of these steps can be improved by applying AI. I mean, so once we call the variance, so I think the next step is, so my question is like, are all these variants which I observe in the sample are functional, are, or like are some of them are like nothing and have nothing to do with the protein, or like I don't know. So that is like one of the challenges in cancer research at gene level. So I think like predicting the mutations which are driving this cancer is one of the important questions to be solved. And there are like lots of methods already in the literature, and the research has been going on from decades to answer this question, and still it's in progress, I would say. So when we look at these methods for driver mutation, so when I say driver mutation, that means the alteration. So I'm like interchangeably using terms like alteration and mutations, both are same. So like the question is like, about what are the driver, what are the alterations that are actually driving this tumor or the cancer in that patient. So to answer that, like there are many methods, and there are most of the machine learning algorithms out there in the literature to answer this question. So I think the general flow of those methods to answer that question is like how. So for most of the supervised learning methods, so what we do, so we feed in the data which we already know the functional labels of those mutations. So I think that there are like some of the mutations from various genes in different cancer types for which we know the functional labels of those from the experiments, right? So we feed in those data to the supervised learning algorithms. We train it with multiple features derived for those mutations, and then we do the feature selection here, and we also do the feature integration and try to find out the best features that define the driver or the. So when I say passenger mutations, the opposite of driver, that means the mutations which doesn't have any effect on protein. So we do this feature selection to identify a driver or a passenger mutation in that particular given cancer. So then we do the prediction. So I think this is one of the important application of AI here at the gene level to identify these mutations. So why is it important to identify the driver mutations? So I think like driver mutation itself is a target for treatment, right? If you know the mutation in particular gene, or a particular gene is carrying this particular driver alteration, so we try to target that gene specifically instead of a general compound that targets the similar proteins. So I think this is the one important question to solve the targeted inhibition, targeted drug discovery, you can say. So any questions here?
Oh, few more questions
Yeah.
are there in the chat box. Yeah, go on.
There are few questions from Naskat Ali. In NGS sequencing and genomes alignment, what will be the anonymously change in the sequencing genomic codon protein?
I'm sorry, can you please come again?
In NGS sequencing and genomes alignment, what will be the anonymously change in the sequencing genomic codon protein? Protein.
So anonymous.
You can read it in the chat box as well.
I think Sunita, the question is not clear. Nazakat, can you reframe the question, please? I think there is some question in the framing part. Maybe he can put it again by changing the word.
There's another question from Pramod Kumar. Apart from cancer, what are other diseases where we can apply artificial intelligence?
I think like, so when I say diagnosis, it need not be only cancer, right? It can be any disease. So any kind of abnormality that can be detected by scans or any kind of imaging techniques, right? So here I'm specifically focusing on cancer. That is because my expertise is cancer research. So Obviously, there are like many other diseases which can be identified by imaging techniques, and where you can apply AI to improve those techniques and to improve the detection of the disease.
I hope this has answered your question. Now there is another question from Ayush Grover. Is there any way that we can predict metastasis? Metastasis.
Yep. I think like one way is again imaging the scans. So when you do the scans, you can also see the different like tumor tissues or the tumor growths across the organ parts which are already metastasis. For example, here I don't think in this scan there is any kind of metastasis because you can only see one tumor. Like for example, if there is metastasis in this particular patient, the scan would be different. Like you can see different abnormal growth of tissues here in the scans at different places. So obviously, if you see more than one, you can call it metastasis, right? So metastasis, it is within the same organ, and sometimes like I think, and the tumor can also metastasize to other organs of the body. For example, lung cancer. So when you scan the lungs, if it is metastasized within the lung organ, you can see multiple abnormalities in the lung scan. So sometimes it can also metastasize to liver. So I think like most of the experienced clinicians here, so they already know, right, like which stage of cancer might have already been metastasized, or based on their knowledge, they would recommend the patient for multiple scans of different organ sites. So from those scans, I think the scan is the first step to identify that metastasis.
So any
Thank
other
you.
question?
Yeah, I have a follow-up question. Is it also possible to understand in which organ the tumor might metastasize to?
So it's like, for example, mostly in lung cancer, we see this metastasis. I think like metastasis is nothing but the progression of the disease, right? So it's like progressing from one part of the body to the other part. So the primary tumor we might observe in the lung, so it can metastasize even within the lung and can go to the other organs. So, and like there are like certain types of cancers where the metastasis is like very common. For example, lung cancer, you could say, and the other cancer is like liver cancer, cholangiocarcinoma, and even sometimes brain. But it's very rare in the brain tumors to metastasize to the other organ sites. So likewise, so it's all again based on knowledge, right? From research, you can answer all of these questions.
Thank you.
Okay, there's another question. Mutation can also be detected through, I think, yes, this question. Mutation can also be detected through NGS data analysis. So are there any accuracy differences between artificial intelligence and NGS data analysis on mutation detection? Is there?
I think like each method have their own, each AI technology or AI algorithm have their own challenges or limitations to detect these variants. So there are like multiple factors in this variant calling, right? It all depends like multiple factors in the experimental setup while they process the samples, like what is the purity of the tumor sample. I think that will affect the variant calling, right? If the tumor sample which I collected is like very pure, obviously I can rely more on the data which is called out from the method. So I think like the major challenge is the data we feed into the algorithms. So the quality of the data is one of the major things which we need to take care to develop an AI algorithm. So yeah, I think like each of these methods have their advantages as well as limitations for it. And I definitely see like when I work with these methods, I see lots of misagreements like between the algorithms. So for example, a method calls a variant, which I might not see in the other methods, right? So this happens. So that is why there's a lot of increment has to be put in to actually take this AI to clinical setup.
You can continue the talk.
Sure. So, I think like right now we are discussing about the driver mutation predictions, the mutations which actually alter, like which actually drive the cancer, right? So, for example, I will just give a brief summary of one of my works during my PhD, how we developed a method to predict these driver mutations. So I think like we developed a gene-specific method, EGFR. So EGFR is an epidermal growth factor receptor. This is one of the prominent driver genes in most of the cancers. So I mean, like you see this particular gene getting altered in many of the cancers. So we developed a machine learning algorithm, which is based on support vector machines. So we fed in 600 single nucleotide, single base alterations into the
Dr.
method.
Anoosha, can you please make it full screen?
Oh, sorry.
Yeah. Thank you.
Welcome. So in this work, so I'm just coming from beginning. So in this work, we developed a machine learning algorithm, which specifically to be applied for EGFR gene to identify driver mutations in that particular gene. So we fed in around like 600 single base alterations. So we only use single base alterations because those are the most prominent ones which we see. We fed in 600 mutations, out of which we used 90% for training the model, and we left the 10% as a blind set. So we did the feature extraction. I think like we extracted around 200 features for those mutations, and we did the feature selection. So out of 200, like around 200, we selected around 20 features to develop the model using 90% of this data. And we achieved an accuracy of 85.4% on the test data set, like 10%. Even on the training data with cross validation, the frequency was like the accuracy was around 80%. So this is one of my experiences with machine learning algorithms. Like it was like hard time for me during my PhD, like jumping around these algorithms, trying to learn them, trying to apply them, and trying to understand the use of it. So that is when I realized like how AI helps in detecting these alterations, which ultimately lead to patient care in the clinical setup. So any questions so far? So after this, like I'll be jumping into the drug discovery part. If not, I can move on.
You can continue.
Okay. So I think like last part of my talk is drug discovery. I think like this is again different domain of cancer research. So when you say drug discovery, so this is again another important aspect of this research. So I think even in drug discovery, it's not as simple as I see right now. So there are like multiple steps within it, or multiple levels of identifying the appropriate drug for a given tumor sample, right? So I think like I will quickly show you an image. So for example, the figure here, the right side of the figure, so each color represents a single patient. And I would say like all of these patients are of same cancer type, say lung cancer. So all of these patients here are of lung cancer, but why did I represent these in different colors? That is because each of these patients, even having similar cancer type, might have different genetics. So because of that, a single, like a common treatment doesn't work to all of these patients. So when we group these patients based on the genetics, like as I did in the right side of the figure, so grouping all the pinks together, all the greens together, so then I try to target the tumors in those patients. So that works better. So this is where like identifying the alterations at gene level are very important. And so I think understanding all of this is an important task for this domain of research, like drug discovery. So different stages of drug discovery are, again, like target identification. So as I said, like you should know, So after the processing of samples, you should know like which particular gene is actually driving the cancer growth in a given patient. So I think that is the target identification. So I think even here, so there are like already a lot of AI methods to understand the association between the target and the disease, between the genes and the disease. So to predict the, for example, for prediction of driver genes, or to predict the network of genes which are important in this in a given cancer. So that is one step, and the other one is compound screening. So I think like we have lots of compounds out there in the databases lying out. So compound screening. So when you say a particular gene is altered in a given cancer sample, I'm not sure like which drug might work for it. So there are already like very well prominent genes like EGFR and FGFR, and like the prominent mutations for which drugs are already designed and are released in the market. But in case if I don't see any of those like prominent alterations in the sample, or if I might see some novel alterations, I don't know like which compound or which drug to give to the patient or which one to suggest to the clinicians, right? So I think there comes a compound screening. So there is again like lots of AI research going on in this field to identify an appropriate compound for a given sample with a given alterations. And then the preclinical stuff. So there you got the clinical trials and all of these to identify the effect of these compound screening and all of these methods, and how the drug response signatures in the given patient, like what kind of response signatures for a particular cancer type. So all of these questions again can be answered with AI. And the other one is a clinical setup where the treatment ultimately goes in, like where all this research ultimately goes in. So there are like multiple steps where there can be benefit, where we can see the benefit of using AI. Any questions here? So
Dr. Anoosha, I think we can go ahead and finish the presentation and then take up the questions.
okay, so I think like I will stop it here with my presentation. So I think like I covered like different parts of cancer research, like starting with the diagnosis. So where AI can be applied and improve that part and fasten the process of diagnosis, which is really important and key to the appropriate treatment. And then the second part is driver mutation or driver gene prediction. So which is again another important thing to give evidence-based treatment to a patient. And the other part is the drug discovery. I don't have any examples here which are ready to present, but like any questions are welcome. So I think like I covered like just 3 of these topics here in cancer research. So briefly to introduce my team. So I think like in all of our previous talks, like Dr. Elson said that there is a very wide gap between how to say the clinicians and the computer scientists, right? There is definitely a gap. I agree to it. But I think like I'm really lucky to be in a particular setup where so I'm a computational biologist, and so our team has like multiple expertise. So we have people from who have experimental background, and we also have people from clinical side. And the head of the lab, he's a oncologist, a physician who looks at cancer patients. So it's like we are like multidisciplinary team together. So I'm really lucky enough to be here. So I think like this communication between the computer scientists and the experimental biologists and the clinicians is a key for this healthcare system. So I think like I will be looking at the patients on daily basis. I'll be interacting with them. So that interaction helps me a lot. So actually that So I'll be seeing patients who benefit from our evidence-based treatment, and that actually fuels my energy daily to do what I'm doing right now. So I think that is really a key for improving the patient care, improving the entire system of patient care. So I will end my presentation here. Thank you.
Thank you, Dr. Anusha. There is 3 more questions. I will read it for you. First is from R. Mithra. During mutation prediction model development, do we need to include gene name and mutation information as feature, or feature information is enough?
I think so. There we can also include like nominal features. I think like when you say gene name or whether it's a driver or not, like those are like nominal features. So I don't think I ever include a gene name into it because it is not needed, because we already do like extract the features of that gene, right? We already feed in all the related features with respect to that gene, and I don't think giving in the gene name would help us. So at least to me, it is not logical to include the gene name.
Okay.
Or it
There's
can
another,
still be nominal feature. Yeah.
there's another question from Mahendra Singh. Which algorithm or method have been used to optimize the SVM model?
Which sorry?
SVM model.
Mhm.
Which algorithm or method have been used to optimize the SVM model?
Oh, there's me SVM model which I developed in my predictions. I think I just optimize the parameters which I use for the kernel distributions. So I just like do the trial and error thing. So I have no idea like which algorithm works on my data, right? Like when I start with it. So I know like SVM is like very well efficient in dividing the data, so discriminating the data, like the linear data at higher exponents. So I think like all that, like I just do the kernel distribution with different parameters and at different dimensions and see how it works. That is how I optimize it.
The question from Anjali. Can drug side effects can be predicted using ML or artificial intelligence, machine learning?
So side effects. So again here, so I think like for any given AI method, the data we feed in is like really important, right? So when you come to the side effects of the drug, so the data or the information is that like we get it from the patients. So I'm not sure like how accurate the data would be. So when we collect it from the patients, that too like when the patients are very sick, so I'm not sure like how reliable it is. So again, so we can always develop a method on any given data, but the thing is like how efficient that could be and how reliable it could be, right?
Thank you, ma'am. There is another question that is, can we use genome and cancer image datasets together to solve cancer-based problems through artificial intelligence? From Mahendra Singh.
Yep. Actually, that's a good idea. So the research has been going on in this particular aspect, combining the genome data with the images. So that would be more reliable, right? Like looking at the images separately and the genomic data separately would definitely add some weight to the predictions. But when you combine like two different aspects of the same unit, when you say like same cancer sample, that is obviously it's like you're adding like more weight to the method, and it's definitely a good idea, and already the research is going on in that direction as well.
Okay, thank you, Dr. Anusha. It's so intuitive to hear from you.